So I would just like to say thank you for inviting me, all the members of the organizing committee, and especially Samantha Weerasinghe, who I've known for a long time as a professor in the chemistry department here. And in fact, in the chemistry department at K-State, we have a rather large collection of Sri Lankan students who've come over the years. And in fact, just recently, I think they now make up over 50% of the graduate students at the university. So we're very proud to have them. We have a problem sometimes with the names, but we do not have a problem with the research that they do. So we're very pleased with what they've done, some excellent research. And we encourage you to think about Kansas State University in the future. So what I would like to talk to you today about is preferential interactions. So my only job here is if you walk away at the end of today and maybe you understand a little bit about preferential interactions, I'll consider this to be a success, right? And we find these all over the place. I put chemistry and biochemistry, but you could add in physics, you could add in chemical engineering as well, and they would crop up all over the place. So, yeah. It's not moving. I should say this is my first trip to Sri Lanka, and I think I'm in tuk-tuk heaven. I was considering actually buying one to go back home, but I think if I drove them the way that apparently I'm supposed to drive them, I would be arrested pretty quickly. But anyway, there you go. So this is just a partial list of some of the research we do in my lab. We're interested in co-solvents. So a co-solvent is anything other than the primary solvent, which is usually water, that you might throw into a solution containing a peptide or protein, and you can manipulate the properties of the peptide or protein. You can change their solubility, you can make the protein fall apart, and this is used a lot in various fields. Well, we're primarily, as a physical chemist, we just like to understand the process and what's going on, right? And so that's what really got me started all those years back at the University of Houston. I'm going to talk a little bit about some of the force fields. We're a computer simulation group, right? So what that means is we try and follow the motion of molecules using the computer, try to learn what they'd like to do. To do that, you have to tell the computer how you think the atoms in the molecules interact with each other. And that's known as the force field, a set of equations and parameters. And they're pretty crappy right now. So in the literature, if you go in, there's lots of nice force fields out there, but I would argue that most of them have serious flaws in them. And so we've been looking at ways to improve those. We'd like to use these things to study peptide protein aggregation and lots of other nice behavior. And we use a theory, which I'm going to just touch on very briefly. Don't get scared. It's not that complicated. To try and basically link our computer simulation data to information on all of these different types of phenomena that we can think of. There's many more, and I'll show you a partial list later. Okay, so preferential interactions. What do I mean by preferential interaction? So if you take a solution, so say you take a beaker, you've got some water in there, you might have another solute, say sodium chloride or something like that. And normally you would characterize that solution by the concentration of sodium chloride that was in the beaker, right? Might be 1 mole or something like that as a chemist speak. And so that tells you the average composition, right? How many sodium chlorides you have, also how many waters you have in a given volume, okay? And that's perfectly fine. But if you were to look at a solution in much more detail, and in particular, if you were to sit on one of the molecules in the solution, you will not see a uniform distribution of the other molecules around that central molecule. Okay? You'll see, so this is what my cartoon's supposed to be showing. I've got a solute a little bit bigger and blue, and the red small dots are my water molecules, right? And this is not a realistic solution, but these are some of the possibilities you might see, right? And we're talking about average composition, like I talked about before, versus local composition around specific molecules. So you can see a situation where basically opposites attract. Right? You've got a solute, and it prefers to be surrounded by other solvent molecules, right? i.e. the solvent preferentially interacts with the solute molecules. So that's an idea of a preferential interaction. or they could be pretty much indifferent to each other, like me and my wife, right? So, no, just joking, just joking. Or you could have the situation where likes attract, where the solute molecules sort of tend to prefer to stick to themselves, and the water molecules tend to prefer to stick to themselves. So at the microscopic level, you see these differences appearing in what looks like a homogeneous solution. Everything's evenly spread out, but it's not, right? And that's crucial because this determines the difference between ideal behavior, honestly, if you're a chemist, you'll know what I mean by that, and real solution behavior. And real solution behavior is what we'd really like to understand, and it's what's important in a lot of processes that we would be interested in. You don't have to be in a solution. You can have a solution up against an interface. And this determines surface tension and absorption of molecules at the interface. And again, you'll see the same kind of patterns. In this case, the blue solute and the red solvent don't occur. They're pretty much evenly distributed. Or the solute might prefer to be at the interface. The solvent stays away from the interface. Or you can have the solvent at the interface and the solute staying away from the interface. So you can manipulate surface tension. You add a salt in, that typically increases the surface tension because the salt likes to stay away from the surface. Okay? It has a negative preferential interaction with the surface. And so we can think about these things, and this has been known for a long time. You can manipulate surface tension. You can change the composition of the solution at the surface, and that can affect the chemistry. Right? So these all have kind of profound. Yeah, okay. The one which I'm most interested in, at least long term, has been when you have biomolecular equilibria. So this is my cartoon of a protein surrounded by a co-solvent and a solvent. This is the native folded, functionally active state of the protein. And you can shift that to an unfolded polymer chain of protein which has no structure and no function by adding in other molecules, these blue molecules. Okay, so you can shift this equilibrium because the blue molecules prefer to interact with the protein in the unfolded form more than they do in the folded form. And this sort of stuff's been well known, but what's been difficult is really how to calculate these things. Yeah. Okay. So these preferential interactions occur all over the place, but how do we calculate them? And specifically for us, how do we calculate them from a simulation and then compare to experimental data to make sure our simulations are doing okay? So we were interested in how they're going to be quantified. So we have this mix match of solute solvent. How do we quantify that? How do we relate it to the thermodynamics, which determines most things that are going to happen in nature? How do we get out of simulations? How can we show that our simulations are accurate, they're reproducing this data? And then how can we use this to understand the nature of solutions in much more detail? So the key is this theory I mentioned before, which is called Kirk-Buff theory. So if you're not a chemist, you can turn off for about 5 minutes while I explain what Kirk-Buff theory is a little bit. Am I doing anything here, or is this? So for those of you who are interested, this is a relatively old theory. The key thing is it's a theory of solutions, and there really aren't very many exact theories of solutions out there. In fact, I would argue there's only 2, which are exact. And the nice thing about it, we can apply it to pretty much anything. It's exact. And what's great for us is that it's great for analyzing computer simulations. The input to the theory is something that comes out of a simulation. If you've ever done one, it's one of the first things you learn to calculate. And I'll get onto this issue of weak binding in just a minute, but it's going to be ideal for studying weak binding. And the bottom line, if you don't remember anything else, is it's a way to relate the thermodynamics of the solution, okay, the thermodynamic properties, to the molecular distributions, the arrangements of the different molecules around each other in solution. That's solute, solvent, 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 solute, solute type of partitioning I talked about at the beginning. So just to try and impress you a little bit, you can apply KB theory to a whole bunch of things, all right? And I won't go through all of these, but if you're a chemist or a physicist, you might be able to spot your favorite little technique. It appears all over the place, right? And so we've been using this to understand lots of things. I'm going to talk a little bit about the effect of co-solvents on chemical equilibria as an example, all right? But there's lots of other things. We can also look at the effect of pressure, osmotic pressure, temperature, and you can do this in any ensemble, i.e. depending whether you're keeping pressure constant or volume constant or so on. Doesn't really matter, okay? So it's a very powerful approach. So before I talk in too much detail about that, I want to introduce you, if you've never seen this before, to the concept of a radial distribution function. So this is a chemist's and physicist's way of thinking about the structure of liquids, right? So normally structure, you think of a crystal, that's got nice structure, but a liquid doesn't really have much structure, but it does have some structure, right? And that's related to those preferential interactions that I introduced before. And the normal way to quantify structure in liquids, and in all the liquid theories that are out, is through what's known as the radial distribution function. And so this is where you sit on a central molecule, you look out radially, hence the radial bit, and then you look at the distribution of the other molecules as a function of distance away from the central molecule. And if you do that and calculate what's known as the radial distribution function, this varies as a function of the distance away from that central atom. You get a distance where no other atom can sit, because it can't sit on top of the central atom. And then you start to see these peaks and valleys eventually going to 1. It goes to 1 because we normalize this distribution by what you'd expect from a random bulk solution, if everything was just randomly distributed. At long distances, it's pretty much random. At short distances, you see lots of peaks. As a chemist, we refer to this as the first solvation shell around our central atom. Okay. Then you might get a second solvation shell, third, and this basically characterizes the structure of our liquid. Okay. And so these are going to be used, these radial distribution functions are going to be used in Kirk-Buff theory. So in Kirk-Buff theory, all we do is we take that radial distribution function, we subtract off 1, that random distribution, and then we integrate over the whole of space, and we call that the Kirk-Buff integral between species I and species J. So if I had a mixture of methanol water, I would have a Kirk-Buff integral corresponding to the distribution of methanol around methanol, methanol around water, and water around water. And this characterizes that distribution. Okay? So these are going to quantify, these integrals are going to quantify what's going on in solution. Here's a typical radial distribution function. If you integrate it, it looks like this. You add up all the negative contributions, positive, negative, positive, till this thing settles down, and then this thing is your Kirk-Buff integral. All right? It includes short range, first shell, and long range deviations from a random distribution in solution. And the key thing is combinations of these Kirk-Buff integrals are going to tell us our thermodynamic properties. i.e., if we know about the distribution in solution, then we know about the thermodynamic properties of the solution. And the only thing you need to remember here is what you can get a quantified number here, but the key thing to remember is if it's greater than 0, we have a net attraction between those two species, and if it's less than 0, we have a net repulsion. Those species like to stay away from each other. Okay, so preferential interaction, preferential exclusion is really what that's telling us. Yeah. So the first example is one of the first studies, not the first study, one of the first studies we did. We wanted to look at co-solvent denaturation of a protein. So we took one of our favorite proteins, lysozyme, and then we looked at urea. Urea is a co-solvent. If you throw it into a solution of lysozyme in water, will tend to unfold the protein. The urea interacts with the protein such that it shifts the equilibrium from the folded form to the unfolded form. And very little is known about that mechanism, so we were interested in using computer simulation to try and understand that. There's a couple of ideas in the literature. There's an indirect mechanism which says that urea just messes up the water structure around the protein, and that's why it shifts the equilibrium. And then there's the direct mechanism, which says that urea binds to certain places on the protein, and that shifts the equilibrium towards the unfolded state. Okay. This is quantified in the literature in a couple of different ways. There's an M-value analysis, which I won't go into. It's totally empirical. Basically, if you plot free energy difference between the folded and unfolded as a function of urea concentration, you usually get a straight line. And so you call M the slope of that straight line. What's more traditional, the binding model approach to understand the effects of urea on the protein. And this, again, tells you about the change in free energy for unfolding the protein in terms of a number of binding sites, and there'll be a difference between the number of binding sites on the unfolded protein and the number of binding sites on the folded protein. And those binding sites are characterized by a binding constant. Okay? And so that was the traditional old worldy way of thinking about protein denaturation. So we come along and we were interested in looking at this using computer simulation. We had that binding model picture, right? There's a number of binding sites on the protein, and there's a binding constant that tells you how tightly urea binds to each of those binding sites. Of course, when we started to do simulations of that, it quickly became apparent that that's a really bad way of thinking about it. And I'll give you some examples of that. So here's, it's not actually lysozyme, but it doesn't matter, a protein in 8 molar urea. And you can see what the first thing you notice here we got the protein obviously in the gray shaded bit. The urea molecules are bigger than the water molecules. It's hard to see them here. But if we just focus on the urea models, the urea molecules, make them a little bit thicker, you'll see that there's a lot of urea in here, right? It's not just a couple of molecules close to the protein. There's a gazillion of them. And there's no way you're going to be able to figure out what all of those molecules are doing. You could say, well, what I'm interested in are the ones which are close to the surface, because they're the ones that are going to be bound to the protein. So let's just strip out all the rest, and this is 5 angstroms away from the surface of the protein, obtained from a simulation. And what you see now is you see ureas which look to be interacting with specific places on the protein. This is a single snapshot from a simulation. Call this take 1. But if you move on in time and then you look at the next snapshot, all the ureas have moved. Right? So what does that tell us? That tells us there aren't specific binding sites for urea around this protein. The urea just smothers the protein and moves gradually all over the protein. And what we really need to be thinking about is the distribution of urea around the protein, not how many binding sites are there and what is the binding constant. You'll never be able to analyze a simulation using that type of approach. So we were interested in changing our attack, right? And there is a third way of thinking about interactions of things like urea with proteins, and that's from an experiment called an equilibrium dialysis experiment, in which you basically stick a protein in a dialysis bag, urea and water can flow in and out of the dialysis bag, and you count how many, or you measure the mass of urea that's entered the dialysis bag. And that gives an idea of the binding, and I put that in quotes just to emphasize that this is not a strong binding to a specific site on the protein. Okay. So this is the situation where we have weak binding, right? Strong binding would be those binding sites, things stick in specific positions. Weak binding, a general smearing of the urea all over the protein. We can quantify that. This is that dialysis experiment gives you the preferential interaction between urea and the protein. This is how they measure in the dialysis experiment to change in masses for each of the components. This is related to chemical potential changes. If you're a chemist, you know that determines the equilibria for a biomolecule. And the nice thing is, using that Kirk theory, all we need to know are these 2 integrals. And this is the integral of the urea around the protein and the integral of the water around the protein. If we can calculate those, we can get back to the experimental data on urea interactions with water. Experimentally, this has been done. Timoshev did this a long time ago. And he came up with a gamma between urea and the native protein of 16, which doesn't mean much right now. It's a little bit pH dependent. Think of that as there's an excess of 16 ureas in the vicinity of a protein over and above what you'd expect just to with respect to a random distribution. Okay? So this is good because now we've got something that we can calculate from a simulation, because we can calculate the Kirk buff integrals. Right? And so we did a simulation of that. And here's our distribution of urea around the protein and water around the protein. And we did this with two different urea force fields, two different descriptions of the interactions of urea with water and the protein. And you see the main features look the same, okay? Except they differ a little bit in the high tier. The OPLS has a little bit stronger, longer range interaction, and therefore depletion in the water interaction. But the main features are the same. Okay. Now the KB, and once you integrate that, of course, then they don't look so similar. And this is that preferential interaction of urea with the protein as a function of distance away from the protein. And again, the first force field, which is the KBFF, gives us flat around 50. The OPLS is really large. And again, to remind you, the experimental is 16. And so this force field, this model for urea, totally gets the binding of urea to the protein wrong. And not just wrong, but an order of magnitude wrong. So we really suck, right, using that. If you use that force field, you're in big trouble. This force field does a little bit better, but it's still overemphasized. So the only way we think to fix these problems is to develop better descriptions of the interactions between the molecules, because that's ultimately what determines the distribution and what determines those integrals. So we've been doing that. We've had a whole program now for a while where we've been trying to improve these descriptions of the interactions between the molecules to get improved force fields for simulations and hopefully correct some of these errors that we're seeing when we look at lots of these phenomena, not just urea denaturation. So I'll do this relatively quickly, but there's two things you need to know about computer simulation normally. If you're doing a simulation, you need to worry about sampling. How often do you observe a particular process occur? That's going to determine the precision of your result. The accuracy of your result is going to be determined by that force field, the description of the interactions between the molecules. So if you have a bad description, it doesn't matter how good your sampling is, you'll have a bad result, okay? So we've been developing lots of these force fields, and I won't go into too many details, but we play around with the parameters that describe the polarity of the molecule, right? How big of a dipole it basically has. And this using the KB approach, we can get a lot more experimental data to compare to, and we can also ensure that we get a good balance of solvation effects, right? We can get that balance between solute-solvent, solute-solute interactions correct. So we do this, maybe I should skip this, but basically KB theory relates these three integrals between the different species in solution to thermodynamic properties. And so as long as you know these thermodynamic properties, these look like complicated expressions, but there's only three unknowns in here. You've got 3 bits of experimental data, so you can extract out those values from experiment. And so we can take the experimental data, get a bunch of experimentally derived KB integrals, take the simulation data, get a bunch of simulated KB integrals, and if we've got a good model, these should match, right? And so we've been doing that for a while now. This is, again, a urea-water mixtures that we've got our KB, and here's the radial distribution functions. Here's the integrated values. We compare with experiment, straight line, crosses our model. This is the other model that we used in a previous simulation of urea, which did a very poor job. You see that the integrals for urea-water mixture are not correct, and so we've got ways to improve that. You can see that in a snapshot. If you take our model for urea-water mixtures, you see it's relatively mixed. There's not any partitioning of regions. You get a high amount of urea or a high amount of water. Whereas if you look at that other force field, you'll see there's gaps where there's water and gaps and places where urea tends to aggregate together. That is all wrong behavior that's in the model, and we think we've got rid of. The nice thing is we've looked at lots of mixtures of different things, methanol in water, sodium chloride in water, and we can get these integrals right. I won't give you the details, but these are some of the other force fields for sodium chloride that don't get these integrals right. So we really do think we're improving the description of the interactions. Hold on. And not just the Kb integrals, but it's important to notice the other properties of the solution. Okay, so this is compressibility, dielectric constant, diffusion of methanol, and diffusion of water as a function of composition, mole fraction of methanol. And you can see we can get these other properties right in addition. So we don't have to sacrifice anything. So that's always good. The last thing I just want to spend a couple of minutes on is, again, what we're working to is developing a protein force field. And a protein's a huge molecule. And so I don't know if any of you know, but the way protein force fields are developed is you break it up into smaller chunks, and you assume that your smaller chunks, when you put them back together, will add up and can be used to describe a protein when they were developed for these smaller molecules in solution. So you might take a molecule like this and split that out, split that out, split that out. These give you different molecules, and then you look at the properties of those molecules. And if you get those right, you basically add them all together, and you hope that the properties of this molecule is just the sum of the properties of the little fragments that you looked at. Okay. And that's been the sort of prevailing thought in the literature for quite a long time, but no one's actually been able to prove that. We think we have a bit of an idea looking at these integrals that we can actually get some confidence that we can do that properly. So here's glycine, and here's NMA, which were two of the fragments that we used before, and we can reproduce these integrals basically in good agreement with the experiment. If you make gly2, so now 2 glycines in a row, and gly3, 3 glycines in a row, where we're adding in NMA as the peptide group each time we go to a bigger glycine, you see that we reproduce the trends that observed at these compositions using that type of approach. So we do indeed think that a protein force field is, to a good approximation, an additive and transferable or it is additive and transferable. And in particular, we see that glycine N aggregation increases with the number of glycines. And so that was a nice result that we think gives us confidence that we can generate a protein force field just by adding up these smaller force fields. So I will finish there. I hope I give you a little bit of an idea of what a preferential interaction is, at least how we're thinking about preferential interaction, and how Kb theory can be used to study those. And we can quantify it, Kb theory being exact. It's good. We're great for computer simulations because calculating a radial distribution function is trivial for a computer simulation. As I said, that's one of the first things you're taught to do. We can improve force fields so we can get better descriptions and therefore more accurate results for our simulations. The only downside that we've seen right now is that because we have this integral over the whole of space, the effects of these molecules tend to be not just of the first solvation effect, which we can normally think of quite easily, like 2 molecules interacting, maybe there's a hydrogen bond or something like that. But now you've got effects over longer ranges, and this is much more difficult to think about, and no one really does know how to do that. So that would be nice to get around, but I don't really know how to do that yet. So I'd like to thank a lot of workers from the group that were involved here. And again, another special thank you to Samantha, who's been a long time collaborator and friend. And thank you for inviting me wherever you are. So, and this has been funded by NIH mainly recently. And I would like to thank you for listening to a non-lightning, boring seminar. So thank you.